This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 295. Recorded on September 7, 2023. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, Vincent and Petra. How is everyone?
Great.
And from St. Louis, Missouri, Petra Levin.
It's great to be here.
Is it really hot in your places?
It's toasty. It's not like it is in New York. We actually have a little bit of a sea breeze, so it's actually quite comfortable, and the humidity is down. But that may all change as Hurricane Lee is on its way.
Oh, wow. Back to back.
Yeah, last week we had our first hurricane adventure, and fortunately Charleston just had a bunch of nuisance flooding, not much damage. But
how about you, Petra? You got hot weather there?
Actually, we don't. It is gorgeous out. It's in the maybe low 70s. It's breezy. It feels a little like fall might be in the air. I know it's kind of a false fall, but it's nice. I can wear a long sleeve shirt outside without being overwhelmed. So it's great. The best of St. Louis weather.
That's right. It's 95 in New York right now.
oh my.
But I like warm weather. It's fine. I'm headed to Salt Lake City today where it's also warm because I was concerned because last week it was snowing. Let's see, Salt Lake.
It was snowing in Lake City last week?
In Snowbird, right?
Oh, not in the city.
A mountain outside, half hour outside. It's 80 degrees in Salt Lake City. And let's see, in Snowbird, temperature, it's 63. Chilly.
Bring a coat. Bring a coat.
It's good you checked.
I wouldn't have checked except for TWiM, so a coat. Yeah, I'm going to have to find. I'll stuff it in my backpack. A coat. I'll bring I think I'll bring one of those puffy things that can squish up into a small volume. How's that? Does that work?
That works.
All right, on to science. We have a snippet and a paper. Don't forget two things first. If you like our work, please support us, microbe.tv/contribute. It doesn't have to be a lot. It could be a coffee a month. I don't know if you can get a coffee for $1 anymore, but you can give us $1 a month. That would be great. And also check out Mark Martin's new podcast, Matters Microbial. I think he's up to episode 5 and talking with various microbiologists. So what does he say? A podcast about the wonders of microbes, microbiology, and microbial centrism. He's doing a good job. So check it out. It's what he's always wanted to do, a podcast. So give him some love. All right. Petra, you have our snippet this week.
I do. And our snippet is a true snippet today. It's a paper in eLife entitled Phage tRNAs Evade tRNA-targeting Host Defenses Through Anticodon Loop Mutations. It's by Dean F. Fontenberg, Baltus A. van der Steen, Anna Rita Costa, and Stan Browns from the Department of Bio-Nanoscience at TU Delft and the Kavli Institute of Nanoscience in Delft and Fagenbank, also in Delft, which is a lovely place to visit if you haven't been there. It's quite beautiful. So this paper, which is really a bioinformatics paper, investigates this phenomenon that's been known for a long time, or this observation that's been around for a long time, but has really confused people. So bacterial viruses or bacteriophage encode tRNAs, so transfer RNAs. They're the little RNAs that carry the amino acids to the ribosome, and they match their anticodon to the codon on the messenger RNA in order to make proteins. So bacterial viruses, like most viruses, really use the host system to replicate themselves. So it's been confusing why they would encode tRNAs when the host is full of tRNAs. So this paper really looks into this, and there have been some ideas about why phage might encode them. Thought they might be there to supplement the host's own set of tRNAs. For example, if, protein synthesis is going full speed and you're starting to run out of a tRNA, the phage needs to make a new phage, maybe bringing some extra isn't a bad idea. The other ideas were maybe the phage encode proteins with rare codons that are not recognized, or the host doesn't have very many tRNAs to recognize those rare codons that would be in the phage genome. But that turns out actually not to be the case. The phage usually match their host in terms of codon usage. And a third idea, which was actually sort of in the same general area as the second one, is that maybe phages DNA and then their mRNA is more rich in G's and C's than the host genome, which would then kind of bias for a certain kind of anticodon in the tRNAs. But that also doesn't seem to be true. The phage DNA tends to match in GC content, the GC content of their host. So in this paper, they have a hypothesis based on the observation that many bacteria have a strategy to inhibit phage infection and they do this by depleting the tRNAs. So if you are a bacteria and you're being infected by a virus that needs your tRNAs, if you get rid of your tRNAs, you might not be able to grow, but you also will make it harder for the phage to grow. And they do this with specialized nucleases, which are directed towards the tRNAs, a subset of which are called anticodon nucleases. And these anticodon nucleases recognize specific anticodons, just like the ribosome mRNA would, but these nucleases actually recognize specific anticodons, and then they cleave tRNAs in a loop of the tRNA. So the tRNA is a kind of intricately folded piece of RNA, and it has a tiny loop right above where the anticodon is. And that's where the nuclease will actually cleave the tRNA. So it recognizes the anticodon, but it cleaves it in this loop. And so they hypothesize that maybe these special tRNAs, that are the tRNAs encoded by the phage, might be there because they're resistant to these nucleases. So it turns out the nucleases only, they recognize specific anticodons, so they only degrade a subset of tRNAs. So maybe the phage are bringing along their own tRNAs because they're resistant to these nucleases, which is a really cool idea. So they go about looking at this. And again, this is all an in silico, as we say, paper. They're using a database. And the other reason, if people have listened to me on this podcast, they know that one of the things I'm very excited about is sort of community science, and especially this course called C Phages, which was developed by Graham Hatfull at Pitt in conjunction with the Howard Hughes Medical Institute. And it's really offered to undergrads, not only in the United States, but around the world. And they originally isolated bacteriophage from the environment, just like the soil. I mean, at WashU, they just go outside and in this class, which is a freshman class, and they would isolate bacterial viruses from the soil that infect Mycobacterium smegmatis, which is a mycobacterium. So if you've heard of tuberculosis, that's also mycobacterium. But smegmatis is not particularly dangerous. I think it can infect people who are immunocompromised, but it's pretty easy to grow and very safe for undergraduates to work with. And these students, undergrads in classes around the world in college, would isolate bacteriophage that would infect smegmatis, this mycobacterium, and they would purify the phage. They'd characterize the phage, they'd sequence them, and some subset of them would get pictures of their phage, which was always, everybody loved that. And they would get to name the phage. And if anyone's interested in C phages, we should put that in the notes to see. But it created, because you have students everywhere, kind of crowd-based science collecting these bacteria viruses, they have this enormous database from the C phages course of bacterial viruses that infect mycobacteria. And we've actually talked about this before as bacterial viruses that have been used in phage therapy to target Mycobacterium abscessus, because there is some crossover. It can target SMEG. Some subset of these phage can also target abscessus, so it can inhibit the growth or kill both species. And it's been used in phage therapy pretty successfully. But here they're just using this database, and we can also link to the website. We'll share that it was generated from all of these phage. There's a paper published in 2017 by Haffel and colleagues that actually just has all the phage that were isolated in it and with their sequences. So they basically, these scientists at Delft, take this resource and they look at the bacterial viruses and their genomes, and they look for tRNAs that are encoded in the genomes. And this turns out to be great kind of resource, because a lot of these viruses that were isolated encode a large number of these specialized tRNAs. Some of them have 30, they say the average is 33 tRNAs per phage. That's quite a few tRNAs per phage. And the hosts that they're using, the Mycobacterium smegmatis, also encode, conveniently, a large number of tRNA nucleases, and a good number of these target tRNAs by matching the anticodon. So they have, a great database, and they have the nucleases. So it's a really good match, and it's easily publicly available. And they basically go through these and different anticodon nucleases, and they do the hypothetical experiment. Will they recognize the phage? And will they be able to cleave that loop near the anticodon? And what they find is that, first, there seems to be a bias. The phage tRNAs seem to avoid encoding tRNAs that are recognized by these anticodon targeting host nucleases. So basically, the host have nucleases that recognize anticodons, and it seems like the phage tRNAs tend to be biased away from the codons recognized by the nucleases. So that's great. They're not even recognized at all. So that works. And then all 10 of the tRNAs that they did find that were targeted by these nucleases had mutations in that loop that would prevent them from being cleaved. So there are a couple things going on there, which I thought was really cool. And there's one figure in this paper. It's kind of all you need to know. It's helpful to read it. It's very nicely written. It's quite easy to read. But this figure is really beautiful because it shows, first of all, kind of where some of these are encoded. And then they look at the frequency with which certain anticodons are present. And then they have this really nice, and because tRNAs are so simple in a way, they have this really nice graphic showing the anticodons and the loops that are recognized by the nucleases, and they compare them to the host, and they can show you that the cleavage sites for these nucleases are often where the mutations in the phage tRNAs are. So basically, it's very easy to see. There's like a little arrow showing, or caret showing, where the cleavage site is and where the cleavage region is. So, right, the nuclease has to recognize that sequence, but it probably is not just recognizing one of the bases. It's probably recognizing the group. And it shows that wherever that nuclease is cutting, there tends to be a mutation in the phage-encoded tRNAs, so it won't be recognized. And I just thought this was so simple and beautiful, and you can just do it by bioinformatics. You don't even have to actually do the experiment. It would be nice to see the experiment, but honestly, I'm pretty satisfied by this. We know a lot about these proteins and how they work and how the phage work, and I thought it was really quite lovely. So they then wanted to know, they only had this, they looked at the mycobacteria, but obviously anywhere where you have enough phages and enough host genomes, you can kind of do the same in silico on the computational experiment. And they actually found, they looked at Salmonella, Enterica, and Shigella flexneri, and E. coli. So those are Enterobacteriaceae, so like the gram-negative bacteria as opposed to the actinomycetes, which is the mycobacteria. And they find the same thing. They find mutations for they seem to have fewer of these nucleases, but in the phage genomes for the phage that infect these gamma proteobacteria, they find that the tRNAs encoded by those phages often have mutations in that loop that are not recognized by the bacterial nuclease, the tRNA nuclease encoded by the host. And I think that's just really nice. They show it in this giant database from the Mycobacterial smegmatis bacteriophage, but then they can probe this less kind of complete data set, but from these Enterobacteriaceae and see the same thing. So it's just another way that the phage can see you come to this sort of ecological equilibrium with the phage trying to avoid the bacterial defenses and the bacteria trying to defend themselves. And so you have two things going on. Obviously, if the bacteria are inhibiting tRNA synthesis, even if the phage supplied the tRNAs, in a way, the bacteria are already shutting down biosynthesis because they can't make proteins very well if they have limited tRNAs. So in a way, they're still have a benefit for having these nucleases, but the phage can at least make their own proteins to get around it to some extent. That's the snippet.
Mother Nature always finds a way.
So we did a paper on TWiM 1035 so there are similar mechanisms in eukaryotic cells. There are anticodon nucleases that are activated upon virus infection. And in this case, they cleave a specific tRNA, not a lot of them, but a specific one that's important for protein synthesis. So again, the host gets shut off as well as preventing viral spread. In that paper, it shows how a pox virus, which is a DNA virus, infects a cell. It activates the nuclease. Not clear how it's activated. But before the nuclease can extensively cleave the anticodon loop, the viral genome produces a protein that binds to the nuclease and inactivates it. So that's the defense.
That's right? An even more elaborate defense. This is just probably natural selection, but that one.
Yeah, so instead of making tRNAs that are resistant, right, you make this protein that binds to it. Those defenses obviously are ancient. They began in bacteria. There are giant viruses, again, that infect eukaryotic cells that encode all the tRNAs. And nobody knows why, but I suspect it's something like this.
Get around the nucleases.
Yeah, there must be many of the hosts of those giant viruses are amoeba. There must be nucleases that are activated on infection, and then maybe the giant virus tRNAs are resistant. So those individuals who work on giant viruses, they ought to read this paper.
Absolutely.
Well, ideas.
The thought I had about the giant viruses is whether or not the giant viruses evolved all these unusual tRNAs, and then the bacterium just threw up its hands, or not the bacterium, the amoeba threw up its hands and said, "Enough, I give up." And it just said, we're not going to change. We're not going to make any more nucleases because of the fitness penalty of making nucleases. Because there is a fitness penalty to making a nuclease. And so they got rid of it, and the virus has no selective pressure to lose the novel tRNAs because it's big, it's giant already, and there's no fitness pressure to get rid of its excess nucleic acid. Whereas the traditional bacteriophage, they have all been about streamlining the volume of nucleic acid that they have in order to fit in the capsid.
Yeah, it's possible. Nobody's looked at it as far as I know.
I mean, I feel like you need to almost do a competition, though, because it seems like the eukaryotic approach is to just shut everything down. And the bacterial approach is also to shut everything down, but then the phage kind of avoid it for themselves, but they can't probably on a global scale and avoid it. So I'm really curious, like, how that would pan out. Like, if you get rid of the nucleases I'm just wondering how much competition you get there.
I think we're witnessing some of that with the evolution of all these spike variants in SARS-CoV-2. It's all about fitness, in the evolution of this virus. And it's really quite remarkable looking at, because, a large fraction of the human population has now either had SARS-CoV-2 or has been vaccinated against SARS-CoV-2 multiple times. And that fitness pressure of just the vaccinated population and what it's exerting on the virus. And that's fundamentally probably why we're seeing all these variants arise.
I think it's all about fitness in the end.
It
in
is.
fact,
it's always about fitness.
It always has been. Some people have always looked at this as a fitness issue, but for poliovirus, we've always thought about neurovirulence,
Mm-hmm.
and people looked for neurovirulence genes, but there aren't any. It's just fitness.
There's no such thing. It's to
just
help
fitness.
the virus replicate. What's the mission of any microbe? Make more.
That's right. And if you replicate for a virus that's in the blood, like polio, if you replicate more, if you're more fit, you're more likely to spill into the brain, which is an accident. It's not part of the normal pathogenesis. And so if you have low fitness, you never get in the brain. So the vaccine strains have low fitness.
Mm-hmm.
But there's always a pressure to regain fitness, right?
Oh, yes.
Because if a virus arises randomly that is more fit, it will outcompete, and then it gets into the CNS.
It gets in.
So when they isolate the vaccine strains, though, from like the sewage, are they actually now more fit, or are they just the vaccine strain?
So
maybe this is a TWiM question too.
No, it's a good question.
It's fair.
Within a few weeks after you're vaccinating, you shed what are called Sabin-like viruses. They're not yet more fit, but after some evolution in the gut, they become more fit, and they can cause. And then they actually, what happens is the vaccine viruses recombine in your gut with other similar enteroviruses and become really fit, and then those can transmit to other people and cause polio if you're not vaccinated.
Okay, so my understanding of polio is that, well, there was a case in New York, right, where someone got it from probably from local, but that one, was that just the Sabin virus that had reinfected, or was that actually one of these ones that had essentially been passaged enough times to become more?
So that was a vaccine-derived poliovirus, which means it's probably recombinant, and it's been passaged extensively through people, and it transmits well, and it's neurovirulent. And so he got it from somebody else who had poor hand hygiene, and since he wasn't vaccinated, he got polio. And now CDC finds those viruses in the sewage in New York.
And those do not look like what they are using as a live vaccine anymore. I mean, they're pretty diverged, I guess is my question. I would love to see how that works.
You can still recognize it as a type 2 by the capsid coding region, right?
Okay.
So the capsid proteins define the serotype, but the rest of the genome is from some gut enteroviruses. You don't even recognize it as a poliovirus anymore, which is kind of scary. It means that you can't use for polio attenuated vaccines because any changes you introduce will be gone. And in fact, there is a more recent, so people tried to improve Sabin vaccines by introducing mutations to lower the error frequency of the polymerase, to lower the recombination frequency of the polymerase. They were all removed by one recombination event, and they're gone.
Yeah, I mean, you can't. So basically, this is a big argument against that vaccine, because the argument for that vaccine is it's easier. You protect the people in your household. You can put it on a sugar, right? This was sort of the argument for using it, especially, not in the US or Western countries, but using it but it seems like now a very terrible idea, and we should only be using the
not really, because on Vincent's special episode of
okay.
he and the leading polio vaccinologist discussed the advantages originally of Sabin, and it was always intended to be used in a vaccine campaign.
Correct.
So that you vaccinate as many people at once to effectively address it, and then everyone is immune and you don't have a naive candidate sitting out there to get the paralytic polio because everybody is immune.
But you have to rely on this being extremely.
It's on the campaign.
Yeah,
and
but the campaign can't be one campaign. It has to be a continuing.
No, it can't be one
campaign.
individual of an isolated case. And
well, that was Sabin's original idea, because he
intent.
knew that it reverted and caused polio, and he said, so the solution is to do massive campaigns, and now they're not doing that anymore. And so they're always, as Michael said, they're always unimmunized people, and they are susceptible to getting vaccine-associated paralytic disease. How did we get on this? Fitness. This is all about fitness.
Well, we see this recombination all the time in E. coli. I mean, as Petra knows better than most, E. coli 0157H7 is only 40% similar to E. coli K12. Its genome is 40% different. 40%. I mean, we're 95% identical to chimpanzees, yet we can tell ourselves apart. But E. coli is still E. coli.
Yeah, well, I don't even know what makes it E. coli. I mean, you compare MG to UTI 89 to 0157, and they're all E. coli, but they're
all different.
Very differently.
Very differently. And, that's the brave new world of why we need microbiologists around to help us understand these very subtle differences.
All right. On to our paper, Michael.
Well, I apologize to the authors because I'm not going to read the 27 or 28 authors here. The first author is Shukla, and the last three authors are Kim Lewis, Tanya Schneider, and Marcus Weingarth, and they're at a number of different laboratories. And the title of today's paper, or manuscript, I should say, is a new antibiotic from an uncultured bacterium binds to an immutable target. So I found today's manuscript during my lunchtime hunt for an appropriate paper for TWiM. What I stumbled upon was the National Library of Medicine is running a pilot to include preprints that result from research funded by the NIH. And these manuscripts are generally posted on bioRxiv. And this particular manuscript has been posted since mid-May. So it hasn't yet gone through full peer review, but it's still worthy our attention because the title is fascinating, because it tells us we got a new antibiotic, the microbe hasn't been cultured before, and it binds to an immutable target. What does immutable mean? And we'll get into that. So I thought I would start today's discussion. Our TWiM colleague, Dr. Dan Griffin, always starts his offerings for his clinical update with a quotation. So I thought it only appropriate that in discussing today's topic, I'd offer the quotation that Selman Waksman used in his Nobel lecture of December 12, 1952, on the occasion of his acceptance of the Nobel Prize for Medicine for the discovery and utilization of streptomycin, another wondrous antibiotic for disease control, notably in the treatment of the Great White Plague or tuberculosis. And here's the quotation that Waksman offered the Nobel audience. "The Lord hath created medicines out of the earth, and he that is wise will not abhor them." And that, of course, is from the Old Testament, Ecclesiastes, chapter 38, also known as the book of Sirach. Well, this paper will describe such a medicine, one that has arisen out of the earth, literally out of soil or dirt. Additionally, this manuscript is not for the faint of heart, as it has some heavy-duty chemistry in it. But fear not, for the discussion puts it all into perspective as to how and why the antibiotic not only does its magic, namely kill microbes, but how it might be possible that the title of this paper should not be considered hyperbole, in that the target of this novel antibiotic is indeed a likely immutable target, in that the target is pyrophosphate, one of the most essential biochemical intermediates in all of biochemistry. So I think it's going to be a fascinating story. First, the authors offered that the uncultured microbial world represents a vast, over 99% of all the species that we have sequenced to date and that are unexploited as a source of new natural product scaffolds from which new medicines might be developed. So the first question many of our listeners are probably asking me, what is a natural product scaffold? Well, quite simply, it's nothing more than a guide that offers both chemists and microbiologists insight and clues as to what the molecule or antimicrobial might be doing to impact the growth of the very things they are attempting to control, whether that control be against bacteria, fungi, viruses, or even cancer cells, because, we can isolate anti-cancer medicines out of soils as well. In 2010, Nichols and colleagues developed the I-chip technique, where the I stands for isolation. And one of the senior authors of today's paper, Kim Lewis, was one of the co-authors on that Nichols paper back from 2010. And the I-chip, or isolation chip, is nothing more than a method of culturing bacteria. And using regular methods, we know that about 99% of the bacterial species simply can't be cultured for a variety of reasons, as they don't grow in the conditions that we establish for them in the laboratory. And this problem has been termed the Great Plate Count Anomaly. The I-chip instead cultures bacterial species within their soil matrix themselves, the soil environment. So here's what happens. The soil is diluted in molten agar, and nutrients such that are being made by other microbes in the environment or just diffusing into the local cell's environment have an opportunity to interact with the microbe we're attempting to culture via the I-chip. And the I-chip has a really small compartment, namely a 20 microliter well, hence the term isolation. And what happens is the chip is enclosed in a semi-permeable plastic membrane and buried back into the soil to allow nutrients not available in the lab to effectively diffuse in and supply all the requirements of the uncultured microbe. With this culturing method, about 50 to 60% of the bacterial species are now able to survive. And again, this technology was pioneered by Kim Lewis, Slava Epstein at Northeastern University in Massachusetts. And I'll drop into the show notes some background information on the power of this technique. And there's two really cool articles that I dropped in. One's more science discussion that appeared in Science magazine. And another one is actually of all layouts, I got to remember what I pulled it from. It was from the Foreign Policy magazine, and it does a really great job of talking about this I-chip technology. So if you're interested in it, you can get access to the Foreign Policy article pretty easily, and they go into great detail of how this works. So because of this tool, or the I-chip, antimicrobial miners now have a broad diversity of uncultured bacterial species from which they can ask, are you making anything that might inhibit a medically relevant bacterium, fungus, virus, or even a cancer cell for which I need an appropriate drug to control their growth? By using such an approach as the I-chip, the novel antibiotic Teixobactin, which was published by Ling in 2015. There's a Nature paper also in the show notes about this. It was isolated from the soil bacterium Eleutheria, which simply means from the earth. Now, Eleutheria is a gram-negative microbe that belongs to the beta proteobacter. It's very rare, and it is, again, something that was used where the I-chip allowed it to be isolated. And Teixobactin shows excellent antibacterial activity and has a very interesting chemical structure. it works by blocking cell wall biosynthesis by specific binding to highly conserved lipid precursors. And, lipids are universal in the microbial world. We all need membranes in the microbial world. The mammals need membranes. And so Teixobactin blocks the biosynthesis by specifically blocking to these highly conserved lipid precursors, leading to a formation of a supermolecular structure that then perturb membrane stability. And as we all appreciate, once the membrane is screwed up, the cell pretty much is cooked. Additional antibiotics with novel modes of action using the iChip have also been derived from screening the uncultured bacteria. And one that they mention in their introduction is lasomycin, which is an inhibitor of the ClpP1P2C1 protease, and A. mycobactin, an inhibitor of the public export protein machinery that's universal amongst bacteria and archaea, namely the Sec-Y protein. But this A. mycobactin is specifically targeted against mycobacteria, microbes for which we don't have a lot of really great antibiotics. In fact, Waxman was one of the last individuals to isolate a great antibiotic against the mycobacterium, and that was, of course, streptomycin. Well, this paper is reporting on an iChip screen of an uncultured bacterium where they described the characterization of a novel antibiotic that they named clovibactam, a molecule that has a novel mode of antimicrobial activity, and this is the key, without any detectable resistance. Now, the authors go into great lengths describing how they encourage the uncultured to grow, and they first start by telling us that spores and environmental isolates also need some nurturing and prolonged incubation and to initiate growth. And they sort of did themselves a favor, but by first incubating their soil specimens at 65 degrees centigrade for 30 minutes in order to take out the mesophils, whereupon they then diluted the soil extracts to extinction to make their makeshift microtiter iChip plate, if you will, to achieve a final theoretical density that would result in approximately less than one bacterial cell per 20 microliter well. So they're effectively really pulling out that dilution, and you have about 20 microliters of a soil slurry that then is contained in this agar matrix that they slap a semi-permeable membrane over, and then they stick the sucker back into the soil that they got it from, and they bathe it. And then very cleverly, they look at this and they effectively follow this. And this process is by no means fast. They monitor these microtiter-like wells under a dissecting microscope at 50x at 1, 2, 4, 8, 12, and 16 weeks. So this isn't your overnight and you're done. This takes some commitment to continue to monitor your screens. And then when they see a colony, they attempt to subculture it out of these microtiter plates, and they subsequently then screen them for antimicrobial activities on a nutrient agar plate that's overlaid with Staph aureus. And if the isolate that they just selected out of the soil is making an antimicrobial that is effective against its target, namely in this case Staph aureus, you're going to see an inhibition of Staph aureus from its ability to grow. Again, it's the equivalent, if you will, of a backwards plaque assay, where the microbe is making an antimicrobial rather than putting a phage that's causing a hole in the agar. The microbe is making an antibiotic that is not allowing the Staph aureus to effectively grow or form a lawn. One of the microbes that was producing an antimicrobial that was detected after 12 weeks of incubation, and then based on its DNA sequence, was found to be 99% identical to Escherichia terrier, that they originally isolated that texobactin from back in 2015. So this is, again, the microbe that means free from Earth. It's a gram-negative that belongs to the beta proteobacter. It's very rare. Not only did this microbe produce one antibiotic, but it was actually producing two. The first antibiotic that it was producing was calimanticin. And to make the long story short, the authors attempted to increase the metabolic flux of this novel compound's production simply by isolating and then disrupting the genes responsible for the calimanticin production. And because we're in the era now of whole genome sequences, we have gone a long way from the time of Selman Waksman, where he was trying to discover the operon that would make streptomycin. Today, we just do whole genome sequencing. They were able to identify the biosynthetic gene cluster with 55% identity to the calimanticin botulinum operon, and they were then able to reduce production of calimanticin below detectable levels by interrupting the first gene in the operon, the BAT1 gene, using homologous recombination through a suicide vector. And as I'm reading that, I'm scratching my head, why did they do this? And the answer is they wanted to look at the fermentation, liquor, or broth of Phageria carolina. The reason they named it substrain Carolina is because the soil was from North Carolina. They then separated that broth or fermentation liquor by HPLC. And the same bioassay, they were able to subsequently identify a second antimicrobial, a novel depsipeptide. I warned you that there would be some chemistry, but fear not. A depsipeptide is nothing more than a peptide in one or more of its amides. The amide group or the amino group is replaced by the corresponding ester. And I'll drop a structure into the show notes so you can quickly review your organic chemistry to look at that. And then I'm going to actually put in the structure of the depsipeptide that they isolated, clovibacter.
I think it's worth noting also this, when you're talking about the structure, these things aren't made in like one shot by one enzyme, right?
No.
They have on figure 1, if people want to see the synthesis, it's amazing because they're made by these giant, even the ones that Waksman was isolating, these giant enzymes and enzyme sets. It's basically called the biosynthetic gene cluster, and it's like an assembly line for these, and they have all the steps in it, which is just cool to see. Sorry, I just wanted to make sure people.
No, so any of you taking organic chemistry, just imagine getting this on the final exam. Please synthesize clovibactin for me. Now, the microbes have figured this out, and this molecule displays 2 D amino acids, and the pop quiz, D amino acids aren't found in proteins. In its linear amino terminus, in an uncommon residue, D3 hydroxyasparagine in the Depsy cycle. Now, this wondrous molecule that they isolated from this genetic cluster has antibacterial activity against a broad range of gram-positive pathogens, including, and this is a long list of the who's who of bad infections. First up, we got MRSA, methicillin-resistant Staph aureus. Then we got daptomycin-resistant Staph aureus. And daptomycin is an expensive antibiotic. It's about $1,000 a day, as well as the vancomycin intermediate-resistant Staph aureus strains, also known as Visa strains. And one of the most challenging microbes to treat, God forbid you get this in a hospital, is vancomycin-resistant Enterococcus faecalis. And often that can cause endocarditis. And if you get that, you better get your affairs in order because it's quite frightening. So VRE is very scary. And this drug that they've isolated, clovibactin, works against these multi-drug resistant creatures. The data that they offer convincingly shows, and this is in figure 2A, that clovibactin is bactericidal. And they present this very nicely on a semi-log plot. They're measuring colony forming units per mil. So they're growing the Staph aureus, and they're showing that indeed within 25 hours, you basically are in the weeds down at 10 to the 1 microbes, which is 10. So you're at about the limit of detection of good old Petri plates. And, they comment that clovibactin is bactericidal. Staph aureus, the minimum bactericidal activity is about 2 times the minimum inhibitory concentration. And then they examine the time-dependent killing in detail, which is 2A. And what they commented on is clovibactin was more effective in killing Staph aureus as compared to vancomycin, which often is the first line of defense antibiotic when treating things like methicillin-resistant Staph aureus or MRSA. They also notice that clovibactin produce unusually strong lysis of the cell culture, and they quantified this effect. Now, figure 2B shows you what it looks like in a microtiter plate, and it's a little hard to see unless you really blow up the figure to see the reduction in turbidity, and lysis means a reduction in turbidity. But then in 2C, they give you a spectrophotometric way, and you can quickly see that clovibactin really does significantly lyse the bacterial cell, much better than vancomycin. Vancomycin is in the weeds. Now, they also add teixobactin, the drug that Altaria originally elevated from their initial screen in 2015. And tecnobactin also causes significant lysis, and they've worked out the mechanism of how tectobactin works, and it is able to produce this rapid bactericidal activity and induce lysis, and it's mediated by the gene product of the ATLA gene in Staph aureus, whose activity is really the major cell autolysin of Staph aureus. So it's what the organism has to facilitate cell division, and it effectively interacts with that. Now, cleverly, to investigate the contribution of this gene product, ATLA, to the activity of clovibactin, they get a mutant of Staph aureus without the ATLA gene and simply ask if they still see lysis. And the antibacterial activity against the delta ATLA deletion mutant was examined and compared to wild type. And clovibactin at the 2x MIC is still induced strong lysis, more pronounced than tecobactin. And you can see the mutant experiment in panel 2C. And strikingly, the lytic events were only slightly affected by the deletion of the delta ATLA mutant, suggesting that clovibactin-induced lysis does not primarily, like the teixobactin, rely on the ATLA activity. So that was pretty cool. But it also told them that the observation of this pronounced lysis may be akin to something that is typically observed when we wash our hands with a detergent, which are effectively killing the gram-positives by rapidly destroying the cell membrane. But lysis was not induced by the rapid production of pores in the membrane, as evidenced by an absence of potassium efflux from staphylococcal cells, in contrast to the pore-forming antibiotic, nisin, which makes pores. And then they asked whether or not clovibactin treatment could lead to rapid membrane depolarization. And any of you who have played with live dead cell staining, that's effectively what they're doing. They're doing live dead cell staining to ask, is the membrane still energetically intact? And they use 2 dyes, the membrane potential sensitive dye DISC2, or the dye that once membrane depolarization has happened, the dye cytox, which is chromogenically remarkable, will just diffuse in, so the cell will light up. And that offered insight as to whether the membrane was attacked. And indeed, the membrane was still intact.
So that's actually really interesting because they're lysing the cells and yet the membrane's intact. So those are two mutually exclusive ideas.
Activity.
Yeah. Like how do you break a cell without breaking a cell?
Well, then they go down the garden path of Min-D. And so this paper is not for the microbiologically naive, because you have to know about membranes and you have to know how the cell wall is being constructed. Unfortunately, I don't have a sufficient amount of time to go into how cell walls are in nitty-gritty constructed, but the paper does a really nice job doing that. And
I think the only things people need to know for this paper are that the precursors for the cell wall are assembled, the peptide and the sugars are assembled in the cytoplasm. They're then put on something called lipid 2, which makes it kind of is called a cloak. So it hides it and it can get across the plasma membrane. And then when it's outside the plasma membrane, there's a whole bunch of enzymes that link them together to form the cell wall. So there's an inside set of reactions, and then there's an outside set of reactions. And the thing that links them is something called lipid 2.
And bacteria are very clever at effectively not breaching the integrity of the membrane while growing the membrane. They don't actually put a door in. They actually expand the size of the house without ever opening the envelope, if you will, of the wall. So, does this antibiotic work in an infection model? And they take us down this path. Clovibactin was evaluated in a neutropenic mouse thigh infection model with Staph aureus. Using such a model, the mice are treated with cyclophosphamide to disable the animal's immune response. And then the antibiotic was evaluated for its ability to control the infection without the assistance of the immune system. In clovibactin, at a concentration about 30 milligrams per kg of the animal, was better than vancomycin, or they offer that it was comparable. It doesn't look to be statistically significantly different to vancomycin in diminishing the bacterial burden. And again, they're doing log average colony forming units per gram of thigh from the animal. So they're literally grinding up the thigh of the animal and doing a viable comp. So summing up so far, there's a new sheriff in town, this depsipeptide antibiotic clovibactin, that is quite promising in controlling gram-positive infections when contrasted against the standard of care antibiotic vancomycin. So their next step was to identify what clovibactin was targeting. So how do you identify the microbial target of a novel antibiotic? As I cheated in the beginning, I said this is pretty straightforward in today's world of an inexpensive total genome sequencing. And at the same time, you get a second piece of information as to whether or not the drug has a low frequency of resistance, generally a requirement to advancing an antimicrobial candidate for future drug development. You want resistance in a microbial population to be extremely low before you invest any more of the company's money into developing the candidate. So what you do is you attempt to isolate resistance mutants. Simply, you expose a large population of garden variety bacteria, and here they use Staph aureus, and then you compare and contrast any of the resistant colonies that come up to the total genome of the parent strain of the Staph aureus, and you ask, what changed? You compare and contrast from an in silico analysis similar to what Petra described in our snippet. Namely, you ask, what's different? And they can assess what the antimicrobial is targeting, as that is likely where the mutation is actually going to show up if it's impacting one of those steps in the production of cell wall or membrane or protein, whatever it may be. The only problem was when they plated Staph aureus on media containing clovibactin, even at low concentrations, namely 4 times the minimum inhibitory concentration, no resistant mutants were produced, leading them to suspect that the frequency of resistance was approximately 1 in 10 billion cells, or at a frequency of 10 to the minus 10. So since the antibiotic was not cooperating and giving them mutants, that was good news for drug development, but it was bad news for them about what's the target. They then asked the question about the potential biosynthetic pathway that clovibactin might inhibit. So in order to accomplish this experiment, they incorporated the labeled precursors into the major biosynthetic pathways, whether it be DNA. So what do you use for DNA? You use radioactive phosphate. Vincent glows in the dark still from all the P32 he used in the era of his days in the lab.
Short half-life. Short half-life.
Short half-life. That's true. And then there's RNA. Protein, of course, is radioactive amino acids. And then, of course, peptidoglycan, you have all sorts of other compounds that they may use. What they learned by asking what radioactive precursor was not incorporated in the presence of clovibactin, they learned that clovibactin interfered with the incorporation of radio-labeled N-acetylglucosamine. And of course, N-acetylglucosamine, as any first-year microbiology student will tell you, it's N-acetylglucosamine and N-acetylglutamic acid that serve as the building blocks of peptidoglycan. So this drug is impacting cell wall biosynthesis, which is a really great antibiotic to have because eukaryotic cells don't have peptidoglycan. So it's a really great antibiotic because it's not going to have off-target effects on our cells. Now, their third figure principally confers that clovibactin targets cell wall biosynthesis. In the experiments that they present, they precisely identify the targeting steps, where in panel 3F, they offered that clovibactin inhibits those membrane-associated steps as the precursors are brought out of the cytoplasm and rotated through the grease. It inhibits the membrane-associated steps of peptidoglycan and wall teichoic acids, or WTA synthesis, in vitro, concluding that the clovibactin inhibit a cell wall biosynthesis reaction that consumed lipid 1, lipid 2, lipid 3, and the wall teichoic acids, or the undecaprel pyrophosphate, as a substrate in a dose-dependent manner, suggesting binding to these lipid intermediates rather than inhibiting enzyme function. Confirming this result, they added the addition of purified cell wall lipid intermediates, and they were able to antagonize the antimicrobial activity of clovibactin and then simply restored growth. It's again, you add back precursors to effectively suck up the clovibactin, and that's found in figure 3G. So again, summing up so far, because this is a complicated story, clovibactin binds to the pyrophosphate moiety of multiple essential cell wall precursors, the C55 pyrophosphate, the lipid 2, and the lipid 3 wall teichoic acid, from different cell wall biosynthetic pathways. And the clovibactin, in a series of hardcore chemistry experiments, binds to the pyrophosphate moiety of these precursors. So from the external medium, clovibactin settles at the membrane surface where it binds these cell wall precursors as they're beginning to stick their head up, if you will. The leucine side chain on the N-terminus helps partition the clovibactin into the membrane. And remember, leucine is effectively a relatively hydrophobic amino acid, so it's going to get in there. And the backbone amino protons of clovibactin's Depsi cycle then directly coordinate with the pyrophosphate, while the hydrophobic Depsi cycle side chains, the alanine, the leucine, and the leucine 8, so alanine 6, leucine 7, and leucine 8, surround the pyrophosphate group like an adjustable glove, which is a very unusual reaction they offer, which is presumably entropically favorable. And this is where the chemistry leaves me, but I'll just quote them: by replacing boundary water, which then facilitates or enables efficient binding to distinct indispensable cell wall precursors. So we are effectively going after all of the precursors that the cell needs in order to build its wall. By Clovibacter attacking this highly polar target like the pyrophosphate with the hydrophobic warhead of the leucine, this was such a striking counterintuitive mode of action, and such selective binding to the pyrophosphate moiety of lipid precursors is achieved by the formation of what they term a supermolecular complex in this subsequent oligomerization into an irreversible higher order fibril assembly of interacting clovibactin molecules bound to targets on the surface membrane. So I'm going to leave you read that. The moral of the story is clovibactin is targeting the pyrophosphate of the cell wall precursor. And pyrophosphate, from the earliest days of biochemistry, everybody has seen it over and over in all the biochemical reactions. This is a straightforward immutable target because you can't swap out pyrophosphate. It's too important in the overall enzymology of the cell.
Pyrophosphate is everything for cells.
It's everything.
ATP and GTP. It's everything.
It's everything. So the moral of the story is, if you want a compound that will have a long shelf life as an antimicrobial, make certain to target to something that the cell can't live without. And pyrophosphate is such that molecule. And I apologize to the authors for not going into their fibril biochemistry, but it's extremely elegant, and I knew I couldn't do it justice in the time I had. But this is an especially cool paper. I'm looking forward to seeing it in print, and I'm looking more forward to clovibactin getting into the market. But as they point out, it's going to be a heavy lift to get this antibiotic to the market.
Why is that?
Cost. They're estimating billions of. They didn't comment on how much it costs, but when you read about bringing a new antimicrobial to market, the foreign policy article talks about the cost of bringing antibiotics to market, and they're quite expensive.
Yeah.
And big pharma would much rather develop the next Lipitor than the next antimicrobial, which you only take for 5
I think
days.
there's some other issues with this one too. Well, first of all, this paper is really interesting because it's literally written chronologically, whereas most papers, which makes it a bit challenging to read, you almost should just go to the last figure, if you want to get to the point, because, there's so many experiments that we do generally that are important, but you don't necessarily want to put them in chronological order because it makes it more challenging to convey your conclusions. So I thought that was interesting. The other thing about this is because what it's targeting is going to be every bacteria everywhere has this thing, and it's important for every aspect of cell, not every aspect, but major aspects of cell envelope synthesis. It's not a very specific antibiotic, so it would fall into sort of the Cipro category. And because pyrophosphate's involved, I'm actually not sure. It's probably going to have some off-target effects that might be problematic. So
we have to look at the toxicology for sure.
I think there are many, and in addition to the cost, I think actually those three things are actually equally concerning as an antibiotic. But I like that they were able, because this iChip was developed a while ago, And I'm glad to see that it's bearing fruit because, now you can mine for antibiotics in ways that we couldn't before. So it's really promising to see this.
So is it possible that we'll have side effects that are not anticipated in people because it's hitting pyrophosphate?
Well, the thing is that it's probably not going to be active against the gram negatives because they have that outer membrane. And it probably won't work against tuberculosis because it's got the mycolic acid grease layer because the mycobacteria are diderms as well. But it will work against the gram positives. And again, you worry about it, whether or not it could get into places it doesn't belong, especially if you use a large volume of it.
I mean, if it gets across, it probably, does it get across
to our cells?
Yeah, I mean, to our cells. I mean, the outer membrane maybe, but I mean, the first thing I would like to see is, does it actually damage E. coli? That would be very useful to know. But also, what's its impact on human cells? Because again, pyrophosphate is the engine. I actually just had a conversation with one of my colleagues who teaches biochemistry here, and he spent a whole lecture on pyrophosphate because that's the energy, everything in the cell, right, comes back.
It's pyrophosphate.
I mean, ATP, making nucleotides, it all comes down to that bond. So it's an interesting target, but it's a potentially fraught target.
Interesting. All right. Thank you, Michael. Very, very cool. Both papers very cool today. That'll do it for 295. Show notes, microbe.tv/TWiM. As I said, if you like our work, please support us financially. Microbe.tv/contribute. Questions or comments, TWiM@microbe.tv. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levin's at Washington University in St. Louis. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.